Chapter 17 covers why I've been talking about since we started talking about DNA that the code in DNA, the ATCG, is a gene and it makes proteins. Okay, and those proteins are, whatever is needed to do whatever job is needed to be done in the organism. But how does it happen? How does that code lead to a protein? So that's what we're going to talk about in this chapter. Okay, so when you're talking about genetic information, okay, the flow of genes to very specific proteins made from very specific sequence of nucleotides. Again, how does that happen? Okay, the DNA that we inherit leads to specific traits. And again, it's this code, this ATCG is a code to build proteins. But how does that really happen? Okay, proteins again are linked to genotypes. Remember, genotypes is what the genes are, if you have a dominant, recessive, and all that. And then phenotype is actually what you look like. Okay, blue eyes, dark hair, whatever. So the proteins are what are linked to that. Okay, and the gene expression, okay, what you actually see is the process by which DNA, the information from DNA is copied and it directs protein synthesis. Okay, and it happens in two stages. One's called transcription and one's called translation. Transcription, we talked about this a little bit in the very beginning. If you remember, chapter 1 was kind of an overview of everything. Transcription is making a copy of the DNA by the RNA, and translation is actually making the protein from that messenger RNA. Okay, here's an example of what can happen when you get mixed up proteins. Okay, you have an albino. Most of the time, albinos don't survive very well in the wild because they're going to have all kinds of issues. Okay, like an albino snake is great in a zoo, but will not survive in the wild. It would die right away of mutated cells in its skin because it doesn't have anything to protect it. Anyway, back to, okay, so basic principles of transcription and translation. Okay, RNA is the bridge between genes and proteins, okay? DNA is the information. It stays in the nucleus, it's copied, but it actually, if you're talking about doing actual work, it does nothing. What it does is it's the blueprint. You go in, you copy the blueprint, and then you make something from the copies, okay? So the RNA is the copier. The RNA is the one that does all the work, okay? It copies the information from the DNA and then makes the protein, okay? So transcription is the synthesis of RNA. Last chapter, we talked about how DNA makes a copy of itself and it unzips, and then it goes in and fills in the nucleotides. Same thing when a gene unzips, but now instead of DNA nucleotides, it's going to be RNA nucleotides. So it's going to make a copy of the information. So transcription, when you're done with the process, you're going to end up with a strand of messenger RNA. And RNA is not double-stranded. Like DNA is double-stranded. RNA is not, okay? So when you're talking about DNA and RNA, DNA is a double strand. It opens up, the RNA will go in and make a copy of it. But the copy will only be of one part of the DNA. Okay, it doesn't copy the whole thing. Okay, translation then is taking that messenger RNA, using it as a template, and then you have to use some other RNA. You use some ribosomal RNA to help anchor down, and then you have what they call transfer RNA that brings in the amino acids. And again, I'll show you all this in a minute. Well, probably more than a minute. Okay, where it happens, where translation, where the proteins are built is on the ribosomes. Okay, that's their job. The ribosomes are the site of protein synthesis. This is where we make proteins, is on the ribosomes. Okay, in prokaryotes, translation of the messenger RNA, it actually begins before transcription is finished. So it's like it's going in making a copy, and before it is even finished, it starts making a protein. There's not a lot of room in there, so they gotta use the space that they have. But in a eukaryotic cell, transcription occurs in the nucleus. Okay, the messenger RNA goes in and makes a copy of the DNA and then leaves. Okay, it leaves the nucleus and goes out into the cytoplasm. And then the ribosomes, most of them are attached to the endoplasmic reticulum. So it attaches, and then the protein is built. So you got two separate sites, one for transcription, one for translation. In a prokaryote, since they don't have a nucleus, that all happens in the same place. Okay, so eukaryotic RNA transcribes, and then again, it's modified, the process or whatever to get to the finished messenger RNA. In other words, we talked about DNA, we said we have things to proofread it and make sure it's right. Same thing can happen with RNA. Okay, if it copies one of the codes wrong, that's going to be a problem. So it has stuff to fix that as well. Okay. So if you look in a bacteria cell, you know, the DNA, you make little part of the DNA opens up, you make a copy of it. And then from there, it attaches to a ribosome, that messenger RNA, and then it starts making the protein. And a protein is a bunch of little amino acids put together. Okay. The order of the amino acids determines what protein you have. The order of the amino acids is determined by the DNA. Okay. In a eukaryotic cell, what happens is, okay, we have our DNA, we make a copy of it. Okay. And they call it the pre-messenger RNA. And then we reprocess it. We have start and stop codons so that it doesn't keep going. You know, this is where the protein starts and this is where it ends. Because if you don't have those, and it just keeps adding more amino acids and adding more, and then you never get your protein made. So they kind of fix it again. And if there's some issues in here, if there's a code wrong, they'll fix that as well. Okay. And then it's going to leave the nucleus. And that's where translation occurs. Okay. Translation is the actual making of the protein. So it reads. This is kind of like, again, something that goes along and reads. And then the amino acids that match up to the code come and they add that's the order, and that determines your protein. Okay. All right. The primary transcript is the initial RNA. And then again, if you look at the whole process, what happens is you have DNA with its code. RNA comes in, copies it. And then the RNA is what builds the protein. So again, DNA is the brains of the operation, has the information, but it actually doesn't do anything as far as really building the protein. The RNA is the one that does all the work. Okay, so I guess DNA would be the boss, RNA would be the worker. It's the one that actually produces it. Okay, so the genetic code. How are these instructions? I mean, proteins are made from amino acids, but they're very specific. And remember, we talked about, if you just think about photosynthesis and all the different enzymes, each one of them is a different protein, or cellular respiration, all the different proteins that are needed. Those are all made by the instruction in the DNA. So how the heck does that happen? Well, it's the code. Okay. In our bodies, we have 20 amino acids. Okay. And the amino acids have a code. They have a base code. All right. They actually figured them all out. Okay. How many nucleotides correspond to amino acid? 3. Okay. So each amino acid has a code. Like, for instance, if an amino acid is, I think phenylalanine is an example, maybe its code is AAA. So it needs that code to bring its amino acid, or CCG or whatever. They have a code of nitrogen bases that's where they go. They match up. Okay, so there's a triplet code. The gene to the protein. Okay, so you've got this, like I said, you make a copy of the DNA, and the copy is going to be like AATC. Well, it won't be T, sorry, it'll be U, because remember, in RNA, we don't have thymine, we have uracil. But anyway, it has these codes in it, and all they are is the nitrogen bases sticking up. Okay, the words of the gene are transcribed by this 3 nucleotide words of the messenger RNA. So the messenger RNA copies it, and again, it's going to be broken down into groups of 3. And each group of 3 is a code for an amino acid. Okay? The words then are the amino acids. And again, they're translated, but every amino acid has a code of 3. So the code that's on the messenger RNA, let's say the code of the messenger RNA is CCC. It's going to look for an amino acid that has a code of GGG. They're just going to match up like they normally do. So the amino acids have a code, 3 letters, and they're going to match up on the messenger RNA where they belong. Okay, so transcription again, here's your DNA strand. The RNA comes in. So here you see, if I'm going to make a copy of this, I'm going to do the complementary. So A and U, and again, remember, we don't have thymine, we have uracil. And then CC, GG, AA, UU. So this is a complementary code of the top one. Okay. And if you look here, it's going to be the same as this. But how we get it is making a, matching up with that one. So this is my messenger RNA. All right. Each 3 letters is called a codon. It's looking for an anticodon, the match of this, to come up and bring its amino acid. So tryptophan, I think it is. Okay, its code must be ACC. and it comes up here and it matches up and it drops off its amino acid. Now over here we have UUU, so I'm looking for AAA. Comes up here, so the anticodon grabs on here, drops off its amino acid. And so the order of the amino acids depends on the code. And every one of them is just got a 3-letter, okay? So you want whatever matches up to it. So again, transcription is making a copy. The messenger RNA makes a copy of part of the DNA, leaves the nucleus, goes out, attaches to a ribosome, and then it starts these little transfer RNAs start bringing up their amino acids. And each one is specific for, they have a certain code for every amino acid. So during transcription, one of the 2 DNA strands is called the template strand, okay? And it provides the information, the order, okay? It can't do both sides, it just does one side. So it makes a copy of it and out it goes. That's transcription. So transcription is making a copy of the DNA. The messenger RNA makes a copy of the DNA. And remember, when it makes a copy, the one thing that changes is instead of A and T going together, A and U go together, okay? All right, and so the template strand is basically the same strand for the given gene. It's always the same. Whatever that gene it needs, when it makes a copy of it, the messenger RNA is always going to be the same. Okay, so during the translation, the messenger RNA-based triplets, they're called codons, and I just showed you those before. They're read from the 5' to the 3'. Remember, DNA has a 5' and a 3' and then you flip it over to the other side and it's a 3 to the 5. That's their direction, how they fit. Okay, so each codon specifies an amino acid, one of the 20. Now, some amino acids have more than one codon. But again, you don't have like 2 amino acids with the same codon. That never happens. But they do have more than one. And they actually have figured them all out. Okay. Cracking the code. Okay, there's 64 codons that have been deciphered in the mid-1960s. So, quite a few years ago. They figured out, but there's 64 altogether. But there's only 20 amino acids, so some of them double up, actually triple up even. Of the 24 triplets, okay, 64 are for amino acids, and then they actually have 3 that are called stop codons. It's like, you need to quit here, protein's made, okay? If they don't have a stop codon, they're just going to keep adding amino acids and make a huge protein, I guess. So they have 64 or 63 codons for actual amino acids, and then 3 of them are stop. Okay, the genetic code is redundant. Okay, so there's more than one codon for a specific amino acid, but it's not ambiguous. And in other words, if the codon is AAA, it's only one amino acid. Now amino acid can be AAA and CCC or something like that, but it can't have 2 amino acids for AAA. That's what they mean by that. It's always specific for one amino acid. Okay, the codons must be read in the correct groupings. And again, that's on the messenger RNA. So you put them in order, and then you got your protein. Now, if you take off one, you're going to have a problem. If you take off just one nitrogen base, then all the codes get shifted, and then you're going to have a problem. You're not going to get your protein made. Okay, so that's why the code is so important. Here's all of them they figured out. Okay, so here, phenylalanine is an amino acid. It's 2 codons. Here, leucine has 2 codons. Actually, leucine has 5 different codons, if you look. Okay, so all the different amino acids, here's all their codes. And they just went U, U, U. So that's where that comes in. U, U, C. That's how this chart works. But again, these are the codes that produce the amino acids. They figured them all out. And you can see, like, glycine has 1, 2, 3, 4 different codons for it. Okay, valine and whatever. But what you'll never see is like 2 amino acids having the same code. That won't happen. And then here you see your 3 stop codons. When it comes across that, it's like, oh, we're done. Quit. Done. Okay? So this is the chart to show you all of them. Okay, you know what? We're going to quit here because I'm at I only have about 30 seconds left. So we'll quit here, and then we'll pick up on protein synthesis after we're done with the next assignment.